All right. Hi, everybody, and welcome to ABCD-ReproNim Week 13. This is the last Q&A session of the course, and this week we are going to be focusing on Project Week, how it will go, everything to expect for it, and what projects you can join. So we want to thank everybody for submitting your Project Week proposals. We invited everyone who did submit a project proposal to join us live on this Q&A to pitch their project. So that's what we're going to be spending most of our time on today. We had some really amazing projects submitted, so please take a look at the GitHub Issues tab on the project page. And if you don't plan on submitting your own project proposal, then we want you to take a look at the existing ones to see if there's one that you'd like to contribute to. And we'll also be talking about them later so that you can learn more about them. So before we get into that, Jessica Bartley is going to go over what Project Week is going to all be about. We've had lots of questions about, what it's going to look like given the remote format. So last night we emailed everyone a Project Week frequently asked questions document as well as an outline, and Jessica's going to talk about that today. But the last thing that I want to mention before I hand it over to her is that the deadline today is to submit your ABCD data access confirmation. If you do plan to join us for Project Week and haven't provided confirmation to us that you have been approved for access to ABCD data, then you need to go do that as soon as possible. It's located under assignments. Again, it's the ABCD Data Access Confirmation Survey, and you'll upload a screenshot of your permissions so that we can add you to the final list of eligible students for Project Week. Again, deadline is today. So with that, I am going to hand it over to Jessica Bartley, who's going to walk us through Project Week, and she'll be moderating the project pitches portion of this. So take it over, Jess.
Cool, thank you so much, Angie. Okay, so just some quick announcements before we go into some announcements about Project Week. This is the last official day of the course. Congrats on making it all the way to the end. I know there was a lot of material. You guys did great. If you do have any outstanding data exercises that you still need to finish up because you got behind during the course, make sure to work on those and submit them. This is kind of your chance to finish anything that you have didn't have a chance to do over the course. We also are going to be sending out a survey in the next little bit to just gauge how things went for you and see how much you liked the course and what, if anything, we could do better on. This will be important for us in assessing how effective this course was for the next time that we offer it. So please do keep an eye out for that and fill it out when it comes. In preparation for Project Week, we'll be running some maintenance on the Jupyter Hub. So it will be taken down briefly to add some libraries and stuff at the beginning of next week, either Monday or Tuesday. And as we announced in previous emails, we are going to have to remove all individuals who are currently on the Hub who do not have a duck. That's because we're going to be sharing ABCD data across individuals for Project Week. So people who don't have ducks won't be able to stay on the ABCD Jupyter Hub. So if that's the case, please make sure to move any work you have up there off of it and onto your own servers. It was brought up earlier today that I didn't actually tell you how to remove that work. So that's an important part. We'll post in the Slack some commands for you to secure copy them over to your personal machines. We are going to be removing people starting Monday who don't have ducks. So make sure to get on that. And then briefly, before I jump into things, I just want to mention that we'll be holding a GatherTown social hour after this Q&A session. We posted on the NeuroStars and in the Slack the link to join that GatherTown social hour. The password to join is ABCD/ReproNim/course. Okay, so with that, what I want to do is I want to go over a little mini presentation before we go into project pitches that just goes over what Project Week will be. Can you guys see my screen? Give me a thumbs up if you can. Awesome. Okay, so just goes over what Project Week is and what to expect on March 8th. So we're going to talk about who can participate in Project Week, what Project Week will be all about, what to expect, how to prepare, and a schedule of events. And then we're going to get into the real meat of today, which is the project pitches. As Angie said, we have invited a bunch of people who have given a proposal of a project to work on over Project Week to give a little mini 2-minute pitch to kind of inform everybody what their ideas are, what they hope to work on over Project Week. And the goal of this is to foster interest in those projects and for people to join teams of the project that they want to work on. So last night we did send out a Project Week FAQ and a Project Week outline. Those are really good resources to look at for Project Week. So please follow along at home and check those out before Project Week starts. All right, so who can participate in Project Week? Enrolled or active students who have an active ABCD Data Use Certification or DUC. If you don't have a DUC, I am very sorry, but we cannot have you participate in Project Week. It's because ABCD data are restricted, and we are going to be sharing and talking about ABCD data during Project Week. Also, please register if you haven't done so already to participate in Project Week so that we have your info. The last day to register is today. So enrolled students, make sure to complete on Canvas your ABCD Data Access Confirmation Survey. And you can also complete the ABCD Data Access DUC Status Survey to let us know that you plan to take part in Project Week. Also, active observer students, a registration link was emailed to you directly. All right, so Project Week is going to follow a hack week model, which means that it's going to be full of open-ended collaborative project work. This is going to be primarily participant-driven, and there's going to be a lot of peer learning involved. One of the goals of Project Week is to be able to have you source the knowledge of your community and other people who are participating in the Hack Week. We also hope that you'll use this week to build collaborations and honestly have fun. The main objective of the week is we want people to work on something that's scientifically interesting to them in this upcoming Project Week. Okay, so there are two principles of Project Week that I would really like to share moving forward. The first is we hope that people work collaboratively and reproducibly on ABCD related data projects. There are two kind of sub goals in here. One, we hope that people build novel ABCD data analyses that will hopefully turn into publishable manuscripts for you guys moving past Project Week. And then the second goal for this principle is to apply reproducible analysis techniques to work towards project activities and contributions to open source software. During Project Week, you'll have access to some ReproNim instructors on a rolling basis, and this is a really good resource and hopefully a safe space to kind of play around with different reproducible techniques that you've learned about in the course. The second principle of Project Week is to disseminate knowledge broadly by developing an online repository of course and project materials. We hope that by the end of Project Week, teams make their code and Jupyter notebooks, any of the analysis scripts that they built, openly available on a project's GitHub page. Note though that ABCD data cannot be independently redistributed, so make sure that your public repos don't include any GUIDs or any derived data that you can aid in the re-identification of research participants from ABCD. Results though should be shared by returning them back to the NDA collection. Okay, so as you might have expected, projects might take longer to complete than 1 week, and that is totally okay. Think of Project Week as a place to start working on your project and build collaborations that you can carry forward past Project Week. We hope that teams will take the work that they did during Project Week and turn them into reproducible publications moving forward. But the main goal here is to have fun working on projects that you're interested in. When you're thinking about Project Week, what we'd like to see you do is ask yourself what milestones you think you can achieve within a week, and how can you distribute responsibility across the members of your team in order to achieve those milestones. Also, what are the goals that you'd like to get out of the project that you work on? So we have a few computational resources, computational and other resources that we'll have available to you. First, all Project Week participants will have access to the ABCD-ReproNim Jupyter Hub, which is an online server where you'll be able to perform your analyses on the ABCD-ReproNim projects. If you anticipate needing a significant amount of computational power, say anything above processing the neuroimaging data of 50 or more participants, please let us know and we can boost your computational power. We do also have NDA permission to share derivatives of ABCD data with everyone in Project Week. So if you need access to derivatives, let us know what those are and we can add them to the hub for you. Also, we will be using Slack as our main platform for communication across Project Week. One of the things that we want you to do is create separate channels for each project proposal, and that'll be a place to discuss progress and share resources. We'll also be using GatherTown as our primary virtual meeting place during Project Week. The password to join the GatherTown is ABCD-ReproNim-course. You're of course also welcome to use a personal Zoom link if that is something you prefer, or a Jitsi room to touch base with your team during Project Week. Project Week instructors will also be available kind of throughout Project Week. ABCD instructors, the ABCD-ReproNim course organizers and TAs will be available throughout the week to, ask any questions to if you need help or need some insight on something. And we'll also have various ABCD and ReproNim instructors available to support on projects. A timeline for their availability will be announced soon. And then most importantly, a really great resource is your fellow peers during Project Week. One of the goals of Project Week is to have a coordinated access to the collective knowledge of everyone who participated in the ABCD-ReproNim course. So ask questions about things that you don't know. And if you do know a question to something that somebody asked, answer it on the Slack. Crowdsource the knowledge. All right, so for project teams, we will be asking people to work in teams for Project Week. That means that we need at least 2 people per project team. But the good news is you can join more than one project team. So how do you join a team? Well, you go onto the GitHub Projects Repro on the Issues tab, and you comment on one of the projects that you want to work on, or you can reach out to the project pitchers on Slack directly. And what happens if nobody joins your project team? Well, if that happens, if no one has joined your project team by Wednesday of next week, we'd ask you to reach out to us, and we will probably ask you to join another project so that we do have at least 2 people per team. But the hope also is that because you can join more than one team, you will hopefully be able to find at least one other person to work with you on your project. All right, so today what we're going to do is we're going to go over project pitches, which are going to be 2-minute presentations for the 15 projects that we had submitted for Project Week. You can go over kind of offline these projects on the ABCD-ReproNim projects repository under the Issues tab. I do want to note that some of these projects are more fully fleshed out than others, and that is totally okay. The goal of these pitches today is to introduce the ideas and questions that people pose that they want to work on over Project Week. So if you at this point haven't already picked a team or a project that you want to work on, your role for today is to try and identify the project that you most want to work on. And then after today's pitches, please reach out to the project team you'd like to work with by commenting on their GitHub proposal or reaching out to them on Slack. And then after today, we have a whole week before Project Week starts on the 8th, and it's really kind of important that we make use of this interval week so that we can hit the ground running on Project Week. So this next week, what we want to see you do is at the beginning of the week, we want you to choose a project to work on, form your project team. This means creating a Slack channel, starting to have conversations, and we hope that you are able to set up a meeting time with the team that kind of coalesces in this interval week to start developing a plan of what to work on across Project Week. So in that plan, one of the things that we want you to focus on is what is the scope that your team is going to be focusing on during the week? And we want to kind of have you guys build a little bit of a preliminary roadmap and decide on a plan of action and how to distribute some of that workload across the team members. We also would love to see you guys create some concrete milestones that you'd like to reach about your project midweek and by end of week of Project Week. All of this is going to help you create a project plan presentation, which will again be about 2 minutes. And we'll ask that one representative from the team on the Monday of Project Week give a presentation kind of outlining what you hope to do over Project Week, how you hope to achieve the goals that you hope to achieve. I'll send out an email about all of this information so that you have something kind of to look at going into next week. But we will be asking you to email us a draft, and just a draft, by Friday, March 5th that is a week from today, with this information about what you kind of plan to work on project week, how you plan to do it, what the scope is, and any deliverables you hope to have by the end of the week. On that draft that you email us, make sure that you list any members that have already joined your team. Then, on project week, on Monday at noon Eastern Standard Time, we'll do a brief little welcome, and then we'll jump into the project plan presentations. This is where you can elect a delegate from your project team to go over the plan of how you guys plan to accomplish your goals across that week. After those project plan presentations, we will have you go and meet up with your project team to plan how you're going to do the first day and what progress you want to make that day. We'd also like to have you schedule a meeting time across all of your team members to check in daily, just to make sure there's a place to answer any questions and to go over any progress made. Also on Monday, times will be announced on when this will be specifically, but we'll be inviting some ABCD instructors back to be available to answer some of your questions about ABCD data that you'll need to know more about, if there's anything that you need to know more about for working on your projects. And then on Tuesday through Thursday, that's all about the project hacking. This is where you're going to make progress on your plan that you've kind of outlined on Monday. The times for that are going to be determined by each project team, but one thing that we do really want to see is that you check in with your team once a day to report any progress that you have made for that day. And then on the Friday, the last day of project week, March 12th at noon Eastern Standard Time, that's where we're going to be kind of wrapping up the project hacking portion of the week. Find a place to kind of pause on the project or on the progress that you've been making and make a plan of how to move these collaborations forward past project week. Then at 3 p.m., we'll go over some project presentations where, again, a member from each team will make a really brief presentation about what you accomplished during the week. If there's any preliminary results you found, if there were any problems that you had, what you still need to accomplish in order to reach your goals. And that's how we're going to end the week. But we do hope that you carry these collaborations further past project week so that you can hopefully turn them into reproducible publications. And all of this stuff, I know I went through that really fast, but all of this stuff is on the project week outline that was sent out to you guys this afternoon. So do check that out for a more detailed view of everything I went over here. All right, so in summary, kind of, a successful project week should maybe look like having a collaborative project proposal across your team, making a project plan at the initiation of project week, trying out lots of reproducible things during the actual hacking part of project week, version controlling everything, containerizing everything. We hope that this is going to feel like a safe space to kind of ask questions about how to do that stuff and introduce yourself, get more comfortable with working with these reproducible tools. There will be ReproNim instructors available on a rolling basis for you to kind of ask questions to about that. And then at the end of project week, create a final report in the form of a presentation telling us what you accomplished, and hopefully exiting the project week with a GitHub project page where you can share all of the scripts and code that you wrote during the week. I will not spend a lot of time on this slide, but before project week, I just want to ask you guys to go over the ReproNim, the ABCD-ReproNim code of conduct that we've been sending out in all the emails, and give the guidelines for responsible use of ABCD data that Angie mentioned during her JEDI task force talk, I think on week 8, read. So that you can ask yourself if your analyses are really preventing stigmatizing research. Obviously join the ABCD-ReproNim Slack. And just a little plug, because I think they are truly excellent. If you haven't already, take a look at the project week prep data exercises, because I think that they are really excellent resources. And then lastly, just some additional considerations about authorship questions resulting from papers coming out of project week. Please do check our FAQ where we go into some of our answers to the questions that you've submitted about this topic. But we do encourage you to discuss authorship with your team and contribution roles early on in the process. Discuss authorship both at the start of the collaboration and at the end of the project, as contributions often evolve over time. And then also discuss the project with any potential manuscripts with your principal investigators. We do hope and expect you to include your PIs on your project manuscripts, and they'll be able to provide some guidance. And then also the project might end up being more than one paper, and that's okay. So if that's the case, consider how to divide responsibilities across your team appropriately. And the ABCD-ReproNim organizers, all of us, we don't actually anticipate being co-authors on the papers that result from Project Week. You are, of course, welcome to list us in the acknowledgement section if you think that we've helped. But we do hope and ask that you acknowledge the NIH award that made an ABCD-ReproNim possible. Okay, so with that, I want to welcome all of our project pitchers to give a brief 2-minute presentation on what they hope to do over Project Week so that other people can join their teams and help them achieve that goal. So everyone else who's not a project pitcher, this is your time to pick a project team. You can follow along offline at the ABCD-ReproNim projects repository, and we'll be giving feedback on the projects directly via the GitHub projects issues page. I don't think we'll have time to do so on air because we have a lot of project pitches to get through. So with that, I'm going to stop sharing my screen, and I'm going to turn it over to our first project pitcher, which is going to be Lillian Xu. I hope I'm pronouncing your name correctly, Lillian.
And as Lillian gets started, I want to pop in and say we are getting some Q&As that are being submitted. Please go ahead and keep those, keep submitting those during this time. Totally. We'll be answering those offline. We want to prioritize the pitches right now, but we will make sure all of those get answered afterwards.
Absolutely. Okay, Lillian, thanks so much. We can see your screen. You want to give your pitch?
Sure. Thanks, Jessica, for this introduction. I'm Lillian, working with Seth Pollack at University of Wisconsin-Madison, and I want to do, my project is about childhood unpredictability and reward processing. So first, background. Altered reward processing and subsequent maladaptive decision-making is a potential mediator between childhood adversity and later risks, such as psychopathology or problem behaviors. Increasing evidence has suggested that it's not only just the quality of the early environment, but also its pattern that matters in shaping reward processing and decision-making. So for example, inconsistent rewards and punishments, they may change children's reward expectations and lead to different valuations. But given this interest in this rising interest in unpredictability, there's still a lot of ambiguities in how to define and measure this construct. So the first goal of this project is to extract unpredictability from measures of childhood adversity in this ABCD data. I want to combine, I want to do first a hypothesis-driven approach where I will hand select relevant variables, and second, a data-driven approach where I will use regularization for variable selection and PCA or k-mean clusterings for variable clustering. And hopefully I will combine these two methods and create a composite score of unpredictability. And with that score, I will examine whether reward processing in the monetary incentive delay task associated with the childhood unpredictability. So the primary analysis will be done using the baseline data, but it's because the second year follow-up data don't have complete MID data, but hopefully with a repeated data collection, I can develop it into a long-term longitudinal prospective study later on. And if I do see a relationship between reward processing and childhood unpredictability, I would like to see whether the reward processing mediates the relationship between childhood unpredictability and later risks. So these are the broad strokes of my research plan during the project week. If it's too big, I can break it down into smaller goals. Also, I'm looking for collaborators. So if you're interested in this project and if you know how to code in Python or how to analyze neuroimaging data or how to deal with large public data sets, you're more than welcome to join in.
All right, thank you so much, Lily. And we're going to have to cut you off there so that we stay on point.
Sure.
But we welcome everybody else to connect with Lillian if you are interested in this project. Thank you.
Yeah, thank you so much, Lillian. Next, we'll have Catherine Trice. If you could please share your screen, and you can also, if you want, turn your video on.
Yeah. So, hi everyone. Please note while I get the setup that my project has been revised a bit since I posted it. So I will be updating it on the GitHub accordingly afterwards. So statistical learning is the robust human ability to implicitly learn and adapt to regularities from input. In our lab, we have found very strong relationships between statistical learning and reading skills and statistical learning and language skills in adults and children. But there is very little we know about the neurological measures of statistical learning as they relate to language skills and language growth. I hope to use language assessments from the ABC dataset and the ROIs relevant to statistical learning defined by our lab based on a group level analysis of 22 adult learners who have shown robust learning in these tasks to validate the importance of this language network in both concurrent language skills and longitudinal growth along with literacy skills. A secondary goal based on time is to explore the overlap and connectivity between the language network in the brain based on Friedrich et al. 2010 and the statistical learning ROIs. As this topic is rather broad, I'm open to team input on what aspects of the analysis we focus on. My preference is the association between the connectivity of the statistical learning ROIs and the longitudinal language skills, but all these questions interest me. The assessments we would be using would be the picture vocabulary and oral reading recognition tasks from the NIH toolbox available at both baseline and 2 years of age, along with the participants' accompanying MRI data. For connectivity, we'd probably use a resting state connectivity analysis tool such as CON, and for structural, FreeSurfer. I haven't decided on an algorithm yet. I'm actually specifically looking for partners that know more about these different algorithms to help determine the best fit. I'm also open to people's recommendations on the resting state connectivity analysis, as CON is MATLAB based, which I have less experience in, and it isn't always as successful when analyzing high performance clusters. Thank you for your time and interest. I hope someone else is excited about this as I am, and have a good project week.
Thank you so much, Catherine. That does sound exciting, and I'm sure we'll have a lot of people wanting to join your team. Okay, so next, oh shoot, I lost my list. Next, we're going to go with Ali Goldstein, please. Can you share your screen and put your video on if you would like?
Okay, and then, all right, so do you just see the slides? Okay, perfect. So my name is Ali. The current working title of this is Development of Amygdala Functional Connectivity. So this is based off of work I'm doing in my own lab and also inspired by research from Dr. Nim Tottenham. So basically I've summarized the background info on the right, but we know that amygdala functional connectivity with the prefrontal cortex is a primary neural substrate of emotion processing and regulation, which are kind of key to living like a happy and healthy life. We see a switch from positive to negative functional connectivity between these regions in children around 10 years of age, and children who experienced maternal deprivation in early childhood exhibited accelerated development of this like mature negative profile. So as far as the actual project, it's still very flexible. I've listed a bunch of kind of ideas here, but I'd love kind of input from other people who are interested in looking at functional connectivity. But one idea I had was to look at early life experience in the ABCD cohort and sort of explore functional connectivity that way. Another question asks if relationships in middle to late childhood can influence this trajectory the same way we see during early childhood. So sort of getting at this idea if there's like a sensitive window during which this trajectory is flexible. And then finally, maybe even looking at sort of like downstream effects, like how amygdala functional connectivity is related to individual differences in emotion regulation or any other like behavioral measures related to like emotion processing. So as an undergrad, I'm still like kind of new to all of this. So I could definitely use some help with like the neuroimaging analysis. I've run SCA before. with FSL, but would love to kind of understand other methods and software that's out there. All of the ReproNim lectures have been pretty new to me as well. So sort of like coding help with those to make sure they're being implemented in the best way. And then also anyone with like a psych kind of developmental background could be really helpful for identifying assessments and sort of making sure these hypotheses line up with what we expect. But yeah, feel free to get in touch on the GitHub or on the Slack.
Thank you so much, Ali. I think a lot of other people might be in a similar boat for you, so thanks for your presentation. That's an interesting project idea. Okay, we're going to move on to Mohab Yani. If you could share your screen, Mohab. Thank you.
All right, share. Well, hi, everyone. So we are interested in pain. So pain is a problem that can lead to substance misuse and lower quality of life. The problem with pain is it's difficult to quantify because it's a subjective, unpleasant experience. It includes many brain regions, and it also has bidirectional comorbidities. So to measure, there are no measures of pain beyond self-reported pain. So clinicians and researchers usually struggle to diagnose and treat pain, and therefore patients end up suffering more. So the goal of this project is to test whether functional connectivity within and between brain networks that known to be involved in chronic pain is different in adolescents who report pain when compared to adolescents who don't report pain. We also want to know the effect of risk and resilient factors that may interact with the presence of pain on functional connectivity. These factors could be cognitive, psychiatric, physical therapy, and demographics as well. So this research is really guided by previous research on the impact of early life events on functional connectivity in adults, and also on the review of structural and resting state functional connectivity brain imaging in children with chronic pain. The region of interest that we are interested in, the entire brain, but mainly we could boil down them to salience network, sensory motor network, fault mode network, and thalamus and brainstem. So ABCD really year 2 provides really a great measure of pain development and chronic pain in the future. About 36% of the 6,500 youth reported pain. So we want to use mixed effect models to test the extent the association between functional connectivity and presence of pain, and taking into account the interaction between pain and the risk and resilient factors. However, after listening to the machine learning, we wonder whether resting state functional connectivity and resilience and risk factors can be used to discriminate the presence and absence of pain. So we want to create a tree model that uses regression, logistic regression models of resting state function connectome and psychiatric, cognitive, and physical activity and demographic variables in a tree model to see can predict the presence of pain or not in the year 2 data, because this is the only stage where pain was collected.
All right, so, thank you, Mohab. We're gonna have to cut you off there due to time restrictions.
Thank you.
We appreciate your presentation. It looks really exciting.
Thanks.
Yeah, it really does, Mohab, and this is well planned out so far. So I think it's awesome. I think people are probably going to really like this pain study. All right, so we're going to move on to Lauren Hilbowen. Can you please, Mohab, can you stop sharing your screen if you could? Thank you. And Lauren.
Yes, let me get it.
Thank you.
Okay. Alrighty. Hi, everyone. I'm Lauren, and with Jessica Flannery, we've developed a project called Typologies of Temperament, Identifying Adolescent Temperament Profiles that Predict Substance Use Risk Factors. So we know that research is sought to identify early risk and protective factors of psychopathology and substance use to inform targeted treatments and interventions. And one of these factors is temperament, and temperament is a very multidimensional construct. So the intention of this project is to identify temperamental dimensions and typologies within the ABCD cohort using latent profile analysis on the early adolescent temperament questionnaire completed during the 2-year follow-up. And further, this project seeks to examine whether these dimensions and typologies of temperament significantly predict neurocognitive risk factors for psychopathology and substance use. So we came up with 3 main research questions. The first one being, within the ABCD cohort, what are the study-specific temperament dimensions and typologies? And do these temperaments differ, or these dimensions differ by biological sex? And second, do these typologies and profiles parallel prior work, or rather offer a novel solution? And third, are the identified temperamental profiles present during adolescence predictive of neurocognitive risk factors for psychopathology and substance use initiation? So we plan to use R primarily, and within R, the latent profile analysis and logistic regression packages. So anyone that is familiar with R or is interested in different statistical modeling is more than welcome to join in, or as well as anyone interested in kind of these predictive risk factors for psychopathology and substance use. So at the bottom, we're issue in GitHub issue, I believe 10. And then also you can get in contact with me or Jessica Flannery. Our names are in the bottom right corner on Slack.
Thank you so much, Lauren. That is a really interesting project. I hope it turns into some awesome research. All right, let's move on to our next talk, which is Ade Dade-Matthews. If you could share your screen, Ade, and if you want, turn your video on.
Yeah,
cool.
Thank you. Can everyone see my screen? Okay.
Yep.
Yeah, so basically my topic is, my name is Ade Dade-Matthews. My topic is effect of race and socioeconomic status on mental health trajectories of adolescents and older children. And so basically I'm coming from the background that prevalence of mental health problems amongst children and adolescents is high, with estimates suggesting about 10 to 20%, this according to the World Health Organization. And the neurobiological events are considered strongly influenced by socioeconomic status, and these both contribute to neuropsychological development. I also found out socioeconomic status is associated with a wide range of health, cognitive, and social-emotional outcomes in children, with effects beginning prior to birth and continuing into adulthood. So also, cumulative exposure to high levels of cortisol in humans has been related to depression, cognitive deficits, atrophy of brain structures. More so, according to Zhu et al. In 2019, he demonstrated that children exposed to low socioeconomic status environments are more vulnerable to anxiety and altered hypothalamic pituitary axis integrity, and that is reflected by elevated basal cortisol and blunt cortisol awakening response. So the justification is mainly that the relationship between socioeconomic status, race, cortisol, brain structure, and mental health has not been studied together. And the research question is, to what extent does brain structure and or cortisol moderate the relationship between race or socioeconomic status and mental health trajectories? The objectives are to investigate the relationship between race, socioeconomic status, brain structure, and mental health, and to determine if cortisol mediates this relationship. And the basic areas of need, aside from the statistical analysis, which will be a linear mixed effects model and structural equation model to see the direct and indirect relationship of socioeconomic status, cortisol, brain structure, and then subsequently behavior. So I would need help with civet analysis and the structural pipeline for cortisol thickness, volumetric analysis of brain regions, structural equation modeling, and analyzing cortisol data, diurnal or transient changes in cortisol. Thank you.
All right, Adi, I'm going to, oh, great, wonderful. I was going to cut you off, but you ended right on time. Thank you so much.
Yeah.
Really
Thank you, Adi. Yeah, that's a really cool project. I hope people join your team. All right, so we're gonna move on to the next presenter, which is Jessica Deflon. Could you share your screen, Jessica? Thanks.
Yeah. Can you see it?
We can actually see your desktop. Oh yeah, now we can. Great.
Awesome. So hi, my name is Jessica. So I propose a project called a pre-adolescent imaging-based brain parcellation. So the first step of many neuroimaging analysis is to parcellate the brain. What this means is that regions that are similar to each other are aggregated. There are many different ways to define similarity in this case. And here on the slide, I have 3 different examples of parcellations that are frequently used in neuroimaging, but there are many different others. So currently, most studies that use adolescent images use some parcellation that has been extracted from an adult brain. But the underlying assumption is that the parcellation will not change between different ages, but we don't know if this assumption holds for the different age groups. So the main aim of this project is to answer the question, can we create a brain parcellation using data from pre-adolescent subjects? And how similar, or maybe how dissimilar, is this parcellation compared to the adult parcellation? So for this task, I thought about following the analysis that was described by EU 2011 using the ABC dataset. And so there they extract the parcellation using the adult cohort, but we will be using this in the pre-adolescent cohort. And anyone that is interested in knowing more about how parcellation works or has any experience with how mesh parcellation, experience with FreeSurfer or FSLR, has any programming skills, the code currently is in MATLAB. I'd like to put it on Python maybe, but that's something that I'm open to discussion. And if anyone that has or know a lot about the ABC dataset is welcome to join me. And I have listed a few ideas on how I plan to carry this project through on GitHub, but I'm open to any other suggestions. So feel free to join me if you're interested.
Thank you so much, Jessica. All right, let's move on to our next presenter, which is going to be Masha Stoyanova. I hope I pronounced your name correctly, Masha. I think you're on mute, actually.
That's close enough. Thank you. I appreciate it.
Okay, good.
No worries. Okay, so hi, everybody. I'm Maria, but I go by Masha. I'm a doctoral student at American University, and my project proposal is on cerebellar frontal connections in light of language task performance. So the concepts and structures in this presentation are the cerebellum, highlighted in yellow here. This is classically considered an important motor structure, but more recently, it's been shown that it's involved in cognition and specifically language function because it provides feedback and feed-forward models to the cortex. I'm also going to be talking about the frontal cortex, especially the inferior frontal gyrus, which is Broca's area classically involved in language. I'm going to be talking about structural and functional connectivity and language, specifically the perception and production of speech and language-related functions. So my proposal is to establish and compare frontocerebellar functional and structural networks, especially between left inferior frontal gyrus and contralateral cerebellum. So after establishing these networks, I want to discover whether individual differences in reading and language task performance are associated with connectivity between these regions. So this is a novel project because typically finding this connectivity is difficult. Combining functional and structural connectivity makes us more certain about both types of connections and determines if these represent the same thing. And frontocerebellar connectivity is still relatively understudied. And finally, this project can validate previous connectivity findings shown in a variety of papers in much smaller data sets. The details are, I would like to use the baseline data set, not longitudinal, use picture vocabulary, oral reading recognition, and rate auditory verbal learning task, resting state and DTI scans, Tortoise, and R software. I need help with DTI and DWI preprocessing techniques and suggestions for effective corticocerebellar tractography. Thank you.
Thank you so much, Masha. A really interesting project. Sure. All right, let's move on to the next person, which is Amia Carian Castillo. If you could share your screen, Amia. Thank you. I don't know if you are speaking, but you might be on mute, Amia. Yeah, I think you might be muted. If you could unmute yourself.
Sorry, I
No
share my
problem.
screen again. Okay, now you can
Perfect.
see my link. Yeah,
I can
I'm
see
sorry.
you.
Okay,
And we can see your screen. Perfect.
Yeah, I'm presenting this project, which is actually led by Marisa, and is under the issue number 13, and it concerns the shared and distinct neural correlates of reading and attention. So we know that reading disorders and attention disorders are often comorbid, and a lot of studies have focused on their distinct features, but here we want to focus on what is common. So also from the multiple deficit model, we know that there are likely to be multiple risk factors at various levels of analysis, including genetic, environmental, neural, and also behavioral. And on the other hand, we also know that processing speed deficits are often associated with reading and attention disorders, and that processing speed scores can predict single word reading and inattention. So our goal is to investigate distinct and overlapping neural correlates of reading and attention using the ABCD cohort as a dimensional dataset. And these are the 3 research questions that we want to address. First, are there neural correlates of reading and attention in the ABC dataset? And if so, do they overlap or are they distinct? Next, we want to see whether processing speed may moderate the relationship between these neural correlates and reading and attention. And if there is an overlap, can we also detect it at the genetic level? So for the first question, we would use the oral reading and flanker inhibitory control and attention tasks from the NIH toolbox and the resting state functional connectivity. And these are some of the analyses that we propose, but we are open to discuss any other. Then for the second question, we would add the pattern comparison processing test from the NIH toolbox and probably mediation or moderation analysis in a structural equation modeling framework. And for the third, we would add the genotyping data, which is available at the ABCD, and also the twin design, which is inherent to the ABCD study design, to answer questions about like how much do genes contribute to each of these traits and whether there is a genetic overlap that we can measure. So if this is interesting to you, you're very welcome to join. And yeah, I just wanted to highlight here that maybe we are lacking a little bit of knowledge on how to implement these ReproNim practices. But anyone that has any interest is welcome. Thanks.
Thank you so much. All right, moving on to our next one, and a reminder to just try and keep these within 2 minutes so that we have time for everybody. Our next presenter is going to be Bob Benno. If you could share your screen and turn your video on, Bob.
Okay.
Thank you.
Oops. Okay.
There we go. We can see your screen.
Okay, so my project is to look at the potential resilient effects of playing a musical instrument on neural development outcomes in children from low socioeconomic backgrounds. I can't see all this. Okay, so there's studies by Hennessy et al. That shows music engages areas of the brain responsible for executive function and impulse control. And it also strengthens networks and communication. Music is very important, and it's been shown there's a collaboration between the National Institute of Health and the Kennedy Center for the Performing Arts that is going to assess longitudinal studies to assess the outcomes of music interventions on timescales matching developmental trajectories. So I think the NIH is realizing the importance of music in this early brain development, and I see the ABCD study as a particularly rich environment to do that in. Recently, I found a study by Gonzalez et al. That showed that there are associations of low economic status with cortical surface area and poor cognitive performance in the ABC cohort. The data shows that positive economic, psychosocial, and psychophysiological ecologies might ameliorate these risk and developmental outcomes. So I can't see my whole thing here. So the question I have is, can early music education act similarly to the positive proximal outcomes as defined by Gonzalez et al. Upon developmental outcomes? So the variables to be analyzed, I'm going to be looking at the outcome variables which they looked at, such as cognitive performance, which is the fluid comprehension. And that data is available in DEEP, the Data Exploration Analysis Portal, and the total cortical surface area, which is also available. I'm going to use the explanatory variables of socioeconomic status, which can be obtained from DEEP, household income, the baseline data, or as they did in this particular study, looking at the income needs ratio, which requires some coding because it would require looking at numbers of children in the family and whether in the time of COVID, people moved into households, et cetera. So it may be a little more difficult. The music education, I'm going to be looking at the screen of the hours for music. And what I'd like to look at eventually, if DEEP is available at 3.0, is the music and hours per week and a longitudinal analysis. I don't think we're quite there yet. And then finally, I'd like to look at a group factor analysis to examine the relationships between music education and proximal measures such as psychosocial, identified Gonzalez upon brain development. And then finally,
no, I'm sorry, I'm gonna have to cut you off.
Thank you.
Thank you so much for that presentation. This sounds super cool.
Appreciate it.
Just a quick note, all of these presentations are on, or all of these kind of issues are on GitHub with all of the details on it. So go check out GitHub for a complete description of things if we have to cut you off. Sorry about that. But that's a very interesting project, Bob. Thank you for presenting it. I hope people join your team. Okay, moving on to the next person, we have Matt Matoni. Can you share your screen, Matt?
Yeah, is it shared?
Yep, thank you.
Great. So I'm going to be looking at the structural specificity of psychopathology risk conferred by family history. And the reason for this project is we know that children of parents with a parental history of mental disorders are often at a high risk for that disorder. And the previous work in ABCD focused on depression and showed like a family history of depression confers a high risk of depression for the youth and showed that this was associated with some subcortical volume abnormalities in the youth. So my goal is to kind of further on that study by examining like is the risk conferred by a parental history of psychopathology specific to that one disorder or general across disorders? Meaning if like a youth has a parental history of depression, are they just at risk for depression or will they also be at risk for like anxiety, substance use, or other disorders? And then the more novel question is like, are the subcortical abnormalities associated with that parental history specific to that one disorder or would they be similar? So would a youth with a parental history of depression have different or the same neuroanatomical abnormalities to youth with a parental history of like substance use or anxiety? So there's more information on the GitHub, but these are like the main questions I was going to look at and like the main variables I was going to use. I'd be really open to any sort of collaboration, large or small, first year grad student, so definitely appreciate some help. Another thing I was thinking about was using puberty as a covariate and could definitely just use like a print a report for that. But if anyone wants to do like biospecimen work, I think that could be a really cool addition to the project as well. But also like help with modeling and any sorts of like other disorders or certain brain regions would definitely be all ears for other parts of the project and would welcome any collaboration.
Cool, thank you so much, Matt. Interesting project. I hope people join your team because I want to see how it turns out. Okay, next we have Carlos Cardinas. Can you please share your screen? Thank you.
I'm muted. Here we go. Okay, so there's a lot of text on here and you can find all the details on our issue on GitHub. But essentially what we're asking is that we know that cortical gray matter volume is associated with psychopathology broadly. And we also know that there's a lot of findings about white matter tract structure and also white matter diffusion measures, DTI measures, that are also related to psychopathology as well. So one of the questions that we're asking is, there's also this restricted spectrum imaging measure that's also included in ABCD. Which measures water diffusion in a slightly different way to the other two. So what we're trying to see is how do these different RSI measures relate to psychopathology broadly, and also how do they covary or correlate with these other measures of DTI and cortical gray matter volume? So in this very busy slide on the top right, you can kind of see the method that we're going to be using to do this. So we're going to go with either a PLS, partial least squares, or a generalized CCA model in which you're able to compare different blocks of data in this case, we would basically be measuring all these different ROIs from cortical, subcortical, and white matter tracts to these different measures, either DTI, RSI, or just volume, and see how they relate to CBCL syndrome scores. And so what you can do with PLS and CCA is basically in maximizing the covariance between these different blocks or the correlation, you can extract these different design scores or brain scores that you can see at the bottom right. So the idea is that we're trying to see if we can extract latent variables here that we then may be able to then see if other factors are moderating those relationships there. So how can you help out? So our analysis plan is over on the right. We have scripts to do all of this already, but what we'd like to do is be able to implement a lot of the ReproNim things that we've learned. And also a lot of our analysis is done in MATLAB and R right now. MATLAB is not open source. So we would like to be able to implement a lot of these algorithms in Python if possible. We also need help with visualization. So if you want to help us out with any step of this, please join us. We're a good crew of people. Thanks.
Super exciting, and I love your analysis plan and reproducibility goals. Thank you so much, Carlos. Okay, next we have Cassandra Lowe. Could you please share your screen, Cassandra? Perfect.
Really quick, I'll update the GitHub as well. So basically, I want to look at the relationship between white matter tract integrity and impulsivity as measured on the delay discounting task, but specifically looking at the role obesity plays. So the first question is, do we see reduced white matter tract integrity in children with higher BMIs? And is this associated with more impulsive behaviors on the delay discounting task? But also looking at the patterns that SES plays in mediating this relationship, because BMI and SES are actually quite confounded, where we have higher rates of obesity in lower socioeconomic households. This is just kind of an outline of the model, and also looking at the moderating effect of modifiable lifestyle factors such as physical activity, sleep duration, and screen time between SES and white matter tract integrity. And then just to wrap it up, some skills I could use help with is DTI, multi-level SEM. I've only done this once before. In a perfect world, I would like to use generative models to analyze the delay discounting task. And because my pets don't really want to hang out with me, I include some pictures of them because it is the rule, but you have to show the pets on Zoom. And that's
a good rule.
It is the rule.
Thank you so much, Cassandra. We really appreciate it. Interesting project too. I hope people join it. Okay, next we'll have Alexis Brandt. I hope I pronounced your last name correctly.
Thanks, Jessica.
Thanks.
All right, so briefly, I'll share a project that me and my teammates have been working on. It's titled Data-Driven Early Life Adversity Dimensions, Functional Connectivity, and Change in Internalizing and Externalizing Behaviors among Adolescents. So there's a lot of heterogeneity in early life adversity. And so one of our primary goals is to use data reduction techniques such as principal components analysis or factor analysis in order to identify the factor structure of early life adversity within the ABCD sample at baseline. And then to, in turn, try and link these different dimensions of adversity to neurodevelopment, specifically looking at the resting state functional MRI data, and to investigate whether there are differential effects of different types of adversity with different neural networks. And then finally, we want to try and link these processes to the emergence of psychopathology in adolescents and test this within a full mediation model to see whether resting state functional connectivity mediates the association between these dimensions of early life adversity and psychopathology. So if you're interested, feel free to reach out.
Thank you so much, Alexis. I am so excited for some of these projects. They look so cool. Okay, and next and last, we have Morgan Glover. Thanks so much for sharing your screen, Morgan. Go for it.
Ahead of the curve. Hopefully, last but not least, so this is functional connectivity and emotional working memory and anxiety. All of my information, just because we're cutting it close, is on the slide if you need it. And also I will be in the Gather Town for a bit if you want to talk more background, because I'm going to slip right over that. So this is more looking at the emotional NBAC task and then also the mental health protocols. looking at more of a region of interest, maybe in the NBAC task, correlating those with the mental health protocols, specifically more anxiety scores and stress scores, and seeing if there's any kind of relationship in the emotional working memory and stress and anxiety. I've previously performed some contrast analyses and ROI tasks in FSL. Don't have much work in Python or previous ABCD analysis work. And if you have more ideas to expand on this project, feel free to contact me or get a hold of me in the Slack or anything of the above. But if you want more background and anything related specifically to the project, reach out and we can chat, because I would love to.
Thank you so much, Morgan. I think that that's a theme across a lot of the talks of wanting some help with the programming ReproNim aspect of this, and that's what Project Week is for, asking questions and supporting each other in these various needs. I know that was a whirlwind, but I gotta say, I am so excited for all of these projects that you guys just presented. Thank you so much, everybody, for presenting all this stuff. Your job now, for everyone who is listening who has not already joined a project, go forth and join a project. Choose the one or multiple projects that you really want to contribute to and are interested in. And I'll be sending out an email this weekend probably, just giving you some instructions about what to do in this interval week before Project Week. Angie, do you have it? Angie or Dave, do you have any other announcements?
That's it for me. I think you covered it. This has been a really exciting session. It's so nice to finally get a chance to connect about these projects. So we're just really looking forward to seeing what gets accomplished over the next couple of weeks.
I agree. Really great start, and looking forward to an exciting week, or 2 weeks.
All right, thank you all so much, guys. Don't forget, we have our little after Q&A social hour happening in GatherTown right over there. The link has been posted to the Slack and the NeuroStarts, and the password to join is ABCD-ReproNim-course. All right, everyone. Well, thank you so much for all of these awesome presentations, and I'm gonna end it here. Thanks, everybody. Bye.